probably supposed to wait for the dawn good sign, but I think I'm allowed to at least do some advertising before then. So if you're in this room and you're interested in mitochondria and cytokine interactions and all that, I would very much recommend that you show up for the symposium going on tomorrow morning at 9 in Ballroom A. A lot of great speakers lined up that are going to cover topics on this subject. And on a more selfish level, we've got some folks here at the meeting from my lab that are going to be presenting. Most of the focus in our lab is on using plants to study the evolution of cytonuclear interactions, and these folks are going to be talking about some of that work. So Justin Habbard is a postdoc in the lab, will be speaking in the aforementioned symposium. And Kate Rockenbach and Jess Warren are both graduate students who have posters tonight in the poster session. So please go and check out those presentations. I, on the other hand, am not going to be talking about plant work. I'm going to be talking about a little bit of research we've been doing on human genetics recently. And that's somewhat foreign soil for me, not a system I know a whole lot about. I am a human, so that helps a little bit. But other than that, a little bit of a challenge. But what made us sort of start thinking about drawing our attention towards human genetics and away from some of the plant research is this recent debate that's gotten a lot of press in the literature about mitochondrial replacement therapy. So if you're not familiar with this, it's an in vitro fertilization technique that is aimed at women that carry some sort of mitochondrial genome mutation or defect and therefore are at risk to pass that on to their offspring. Involves taking nuclear material from a patient's either egg or fertilized zygote and transferring that onto a donor cytoplasm. And this is real applied medical science. It's a technique that's gone through UK Parliament and has been approved for clinical use, at least through the first stage. And just in the last few months here in the United States, the Institute of Medicine of the National Academy of Science issued a long-awaited report and essentially urged the FDA to move forward with trials and investigations on this technique. So, needless to say, this has generated a fair amount of controversy and discussion. There's certainly ethical issues surrounding this type of technique. A lot of technical concerns and risk factors have been raised. I'm not going to talk about a lot of that. What I am going to talk about is a very interesting evolutionary idea that I think has emerged from this medical debate and been proposed by a number of researchers in some recent articles. And I will try to sort of, perhaps inartfully summarize the argument being made there, and that one concern, one potential risk associated with mitochondrial replacement therapy is in the generation of novel combinations of nuclear and mitochondrial genotypes. So this basically reflects the possibility that you have mitonuclear epistasis, in particular sign epistasis, such that there's certain combinations of matched, if you will, mitochondrial and nuclear genotypes that are co-adapted or co-evolved. And then by generating novel combinations, there's the potential to generate mismatches that might be associated with reduced health or fitness outcomes. So there's a large body of evolutionary literature supporting the existence of these type of mitonuclear interactions, both within and between species across a variety of organisms. I should also point out that because of what's known as mother's curse, there's reason to believe that these effects might disproportionately harm or have a role in male offspring, which is interesting because that recent National Academy of Sciences report actually specifically recommended that initial implementation of this technique be exclusively limited to male offspring. So running very counter to this line of argument that's been proposed. But this is a debate that's not necessarily just about general evolutionary principles. It's specifically about the extent to which these type of mitonuclear incompatibilities could be risk factors in humans, because that's, of course, the target for this type of technique. And in discussing this with some colleagues, specifically Justin Habbard and Peter Fields, who are collaborators on this work, we kind of came to the conclusion that some of the human population genetics in terms of mitonuclear interaction just weren't as well studied as we might like. So what I want to tell you about is a little bit of work that we're doing to address a couple questions. So first, just are there statistical associations between mitochondrial and nuclear alleles in human populations? Or in other words, to what extent is there mitonuclear linkage disequilibrium in humans? The second question is, to the extent that there is mitonuclear LD, can that be described by selection? Can we actually detect footprints of selection for maintaining certain combinations of mitonuclear alleles? Or does it reflect other non-adaptive processes, demography, population structure, etc.? Right, so to try to get at these questions, we've been taking advantage of some published, freely available human population genomic data. Specifically this data set, which includes about 1,000 individuals sampled worldwide, so more than 50 different populations, each genotype at over 500,000 SNPs, and including a large number of mitochondrial SNPs that allowed us to reconstruct mitochondrial haplotype for each individual in the sample. And what we did here with our analysis is just estimate multi-allelic mitonuclear linkage disequilibrium for each individual nuclear locus in the sample set against the inferred mitochondrial haplotypes. And so when we did that, if we just take a sort of bird's eye view of the data, first off, yes, there very much is significant mitonuclear LD at the level of the global human population, not surprising for any organism that has some degree of population structure. So what you're looking at in this plot here are just two different measures of mitonuclear linkage disequilibrium on the x and the y-axis, and we're averaging across all nuclear loci and each of the different human chromosomes, or what each of the points represent. So while the vast majority of SNPs that we looked at showed significant mitonuclear LD, it's worth pointing out that the magnitude of this association is quite low. If you look at the y-axis here and look at R squared, the explanatory power, the predictive power of knowing an individual's mitochondrial haplotype actually does not give you much information about what their nuclear genotype at any given locus is expected to be. Perhaps one of the interesting things, which the red arrow is already coincidentally pointing out for me, is that the X chromosome is particularly high in terms of mitonuclear LD relative to the autosomes. This is probably mostly driven by lower effective population size and therefore higher population structure on the X. But it also might relate to the notion that the X chromosome is preferentially co-transmitted with a mitochondrial genome because of each of their association with female transmission. All right. So that's the easy part, just counting the extent to which mitonuclear LD exists. The much more challenging question to get at, and what we've just started to think about, is to what extent can we explain patterns like this based on selection versus just more demographic processes. And to try to get at that, our premise was, if you're going to expect effects of selection to be shaping particular mitonuclear interactions, those are not going to affect all loci in the nuclear genome equally. In particular, where we would expect those functional interactions to be important, and where we expect that type of epistatic selection to be acting, would be on the subset of nuclear genes that are targeted to the mitochondria, and in particular, those genes that have direct protein-protein interactions with mitochondrial-encoded gene products. So, for example, the classic OXPHOS enzyme complexes, which are at the heart of mitochondrial function. These include, by and large, subunits from both the nuclear and mitochondrial genome. So it's these nuclear genes that we'd expect to show the strongest signatures of selection. So our approach here was simply to divide up our data set, to divide up the genes into background nuclear genes versus genes like this that are targeted to the mitochondrial genome, predicting that mitochondrial targeted genes would have higher levels of mitonuclear linkage disequilibrium if it were driven by selection. When we did that, though, we found no significant difference whatsoever, not even a hint of it. So what I'm showing you here is the mitonuclear LD values mapped across the entire nuclear genome, and all the points in red are mitochondrial targeted genes. We also got a negative result when we looked at the specific set or subset of most directly interacting proteins within the nuclear genome. So this is a lot of data points for a negative result. All right, so back to these two questions that I posed. Are there statistical associations between mitochondrial and nuclear alleles in human populations? Yes, at least when surveyed at the level of global population diversity, but it's important to recognize that those values are sort of relatively low on an absolute scale. And to the extent that we can detect footprints of natural selections shaping those combinations acting in them, this particular test did not provide any specific evidence for that. I mean, I don't think this is a huge surprise, given what we already knew about human population genetics. We're a species with a relatively low long-term effective population size, recent migration out of Africa, relatively low degree of population structure. These are things that are not very conducive to the evolution of tightly co-evolved mitonuclear interactions. This is kind of something we published late last year, but I want to also share some work we've been continuing to do along these lines, and something that was set up fairly nicely by Carl Zimmer's talk on Friday. And that is that the genetic history of humans is rather complicated. And one of the more fascinating recent revelations is the extent to which introgression from other related lineages of hominins has played in that genetic history, particularly Neanderthals. And so a couple important points here that are relevant to what I just discussed. First, all of that introgression is specifically of nuclear genes. To our knowledge, there's no one walking around the planet right now with a Neanderthal-like mitochondrial genome. So all of those introgressed Neanderthal genes have to interact on a modern human mitochondrial background. And that's potentially relevant because the divergence between Neanderthal and modern human mitochondrial DNA far exceeds the amount of divergence between any current modern human mitochondrial haplotypes that exist. So potentially amplifying the strength of the potential interactions or mitonuclear compatibilities that might have occurred. So that led us to think about this question. So did mitonuclear interactions potentially impede the introgression of Neanderthal nuclear genes that are targeted to the mitochondria? All right on time. So this is work also being done in collaboration with Justin Havert as well as Greg No, who's an undergraduate in the lab. And our approach here has been to take advantage of a data set from David Wright's group, where they've done a very nice job characterizing the history of Neanderthal introgression and identifying genes with history of extensive introgression versus genes with very low histories of introgression, and summarizing that as a introgression score. So you can just think of that. Higher scores mean more history of Neanderthal introgression. And our approach has been to apply the exact same logic. Mitonuclear incompatibilities have created an effect here. We would expect that those to disproportionately affect those genes that are targeted to the mitochondria, and therefore we predict that they have lower introgression scores. And when we looked at these data and divided up the genome the same way, that is what we observed. We find genes that are targeted to the mitochondria have significantly lower levels of introgression. This is true both in Asian and in European populations. In light of this question, did mitonuclear interactions impede the introgression of specific Neanderthal nuclear genes? Certainly, we're at early stages here and not at a point where I'm willing to give a definitive yes to that. As you saw, the effect sizes aren't necessarily huge, and those distributions are still very much overlapping. But we are looking at results that are consistent with that initial hypothesis. I started with mitochondrial replacement therapy. Obviously, no one except for perhaps George Church is proposing to do mitochondrial replacement like work with Neanderthal DNA. But I think this is a result that potentially indicates that these type of mitonuclear interactions and compatibilities might have shaped human genome evolution and genome content. So with that, I would like to thank some collaborators. We will provide data as well as funding sources. And thank you guys. Yeah, one quick question, I'm told. Yeah.
Hi. I'm interested you're also able to test for selection. There's arguments, I think there are big brains, these kind of combinations also have functional benefits in their genes, but aren't necessarily due to interactions, but just selection on those genes themselves. Can you test for that?
It's not something we've tested for, but some of those original analyses that say Reich's group's good, it looked for which functional classes might have actually not been impeded, but have been functionally driven by positive selection for integration, and identified a number of things that immune system related genes is one of the classic ones. But that's the work of others, not us.